activation domain will have a specificity, na? So how it will, that part was
No,
not clear to me.
activation domain does not have specificity in terms of target gene.
Okay.
Activation domains, so I would request all of you to think from purely from the perspective of protein-protein interaction. That what is happening? What is happening is that the role of all this complicated thing is one thing and one thing alone, is to recruit the RNA polymerase to the promoter. Now, the DNA binding domain dictates which enhancer element a transcription factor will bind to. Once it is bound, then the activation domain's job is simply to interact with the mediator complex. Now you can imagine that that ability to interact with the mediator complex can vary. Like I will give you example. Say, so the whole biological system, if you carefully consider, works on suboptimal efficiency, meaning you will not have, you will not find proteins natural in the natural setting that are in their most stable conformation, or proteins that bind to a ligand always binds at the highest affinity. It is not like that. It is always suboptimal, because if it is suboptimal, then only there is a scope of increasing it and decreasing it. Regulation can happen both ways. Now, uh, the job of the enhancer bound transcription factors is to interact with the mediator. Now you can imagine a situation where one transcription factor's activation domain is not strong enough to cause a stable association. Maybe you need 2, 3 different kinds. Maybe you need 4, 5 each of 2, 3 different kinds. So that combination therefore gives the maximal ability to interact with the mediator complex. Now, that in that context comes the issue of constitutively active version of transcription factor. That this VP16, the transactivation domain of VP16 presumably has very high affinity towards the mediator complex, that it by itself is good enough to bring the RNA polymerase to the promoter. While in other cases, it is always a combinatorial code. I will talk about it in the, like I have already recorded the third lecture. I have not uploaded it yet, but I will upload it today itself. And then I will upload some more. You will realize that combinatorial code is the thing.
So, like in mouse genetics, also, like you talked about, like we can replace the activation domain by engrel, for transcriptional repression, or by
Yeah,
your lag, sorry, gal4 with for
yeah,
transcriptional.
yeah,
Okay.
yeah, yeah. So
So
that
sequence
these
specificity is provided
kind,
by the DNA binding domain, not the activation domain.
yeah,
Okay,
yeah,
sir.
like where it will bind, that is dictated by the DNA binding domain. And then the activation domain will activate transcription irrespective, as long as it is recruited to the enhancer.
Hmm. Okay.
बस और किसी का कोई सवाल नहीं है?
Good morning, sir.
हाँ, morning.
Sir, I have doubt in like last lectures. Sir, how will you insert the LOXP sites in our desired gene? Hello, sir. Am I audible, sir?
हाँ, समझ में नहीं आया, फिर से बोलो.
Sir,
Repeat. हाँ.
hello, sir. Sir, actually the sound is breaking this side. Sir, how will you insert that IXP sites in our desired gene? Like if there is a 3 exons, we have to knock out the exon 2.
Log sites, sir. He's talking about
Oh,
log
अच्छा,
sound.
अच्छा, अच्छा, अच्छा. It is not that complicated, you know, like uh, okay, let me try to see. Um, those constructs, there is a really bad noise coming out of my laptop. You know, I'm sorry for that. I'm trying to find a good example, okay?
Yes.
Okay, so let me do this. Let me find out a good example from the literature, and then I will get back to you maybe on Wednesday's discussion. Is that okay, Kuna?
Yes, sir. Yes.
Okay, let me do that. Instead of me coming out of with things out of my head, let me find a proper example.
Yes.
Okay. Okay. But few things I observed during the third quiz, which is little worrisome, that uh, there was a question for can you see it? Can you see
Yes,
it?
sir.
Yes, sir. Yes,
Yes,
sir.
sir. Yes.
Okay.
Yes, sir. Yes.
So just after talking about East genetic screen in two, like for such a long time, nobody got this question right. Absolutely nobody. Zero. That is a problem. Okay. Uh, that simply means that your logic is still not developed. And the other thing is
But sir,
that
it's easy, sir. It is the normal, like the first set of East screens, East mutagenesis
ah,
screen.
but 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 the problem is that nobody. And the other problem is that the second question, uh, I was shocked to say the least that almost like I don't now recall, at least 4 out of 6 propose that they will do knockout to figure out if a gene is being expressed there. Like, even if you put a gun to my head, I will not be able to propose that. And the reason is that I have made a mention of blastocyst there. So everybody thought that it must have something to do with knockout. That is where reasoning takes complete leave of absence, and you say something like this. The reason I talked about this, okay, let me see, who can tell me why did I talk about using blastocyst mouse embryo?
Sir, because the mm,
हाँ आयुष बोलो.
Sir, because we don't need to see where these genes are expressed. We only want to see if these genes are expressed in mouse or not. So simply we can take the blastocyst and do RNA in situ hybridization and check if these genes are also expressed in mouse or not.
RNA hybridization is generally done on solid tissue. Blastocyst is too tiny. Why
Sorry,
can't
you are not
do?
audible, sir.
Why? I am not audible.
There is lot of disturbance.
Yeah, like because of this noise that the computer is making.
Can you please
RNA in
repeat?
situ hybridization is not a good way. It should be done by quantitative RT-PCR. Okay.
Okay.
But why blastocyst? Why did I mention blastocyst?
sir, because in the blastocyst, like it will be easier to assess all the genes at a time, because again when it will go into a adult like mouse, then again we have to take the cells from each and every part and see, like in the blastocyst, in one cell all the genes will be in a way active.
That
They
is
have,
exactly the reason.
yeah, they have not undergone the differential expressional what to
If
say.
you go to any differentiated, because you do not know where these 20 genes are, so
Yes.
if you go to any differentiated cell type, you may miss it not because the gene is not expressed in mouse, you will miss it just because you looked at the wrong place.
Yes, yes.
So that is the problem I saw repeatedly. Um, and what are the other issues that you have as a class from the chapters we have already covered?
So, like in our slot, we could not, that protein blast. We all answered that nucleotide blast, so that was
Ah,
also,
so that
yeah,
is not just your slot, that is in
yes,
every slot.
yeah,
In 2 exams, okay? Those I explained the rationale for doing a protein blast in the second exam, they could come up with the right answer in the third exam, but that was only 2, 3. Majority completely missed it, uh, and that is because you are not thinking. Only knowing the keywords does not help you to become a scientist. You need to know the utilization of each and every technique. Or coach,
The gray mouse generated.
Free mouse generation.
Like how do we create the three mouse for our
Knock in,
specific
knock in,
application?
knock in.
So
Knock in
see,
or transient
free
approach.
free knocking is rather simple. That I can show you right away. Can you see?
Yes.
Yes, sir.
Okay. So you see the green box. Hello. कैसे लगा? You see this green box? This green box can be anything, right? Let us say the green box is green. Okay. Now you see where the homology arms are. Okay. And imagine that this is where the promoter enhancer is.
Yes, sir.
Now you see the neo cassette that will give you selection. This is the other gene that you are trying to express. Let us say in this case it is Cree. This is the other homology cassette, and this is the counter selection cassette. So upon integration, this will look like this. Now you see very interestingly here, the new gene itself is flanked by lox peptides.
Yeah, yes.
That is for a technical reason. So let me tell you why it is done. Just a minute. So it is done because the neo cassette has not been redeveloped. If you read the literature, it is always said to be PGK neo. PGK neo means phosphoglycerokinase neo cassette. Now this phosphoglycerokinase enhancer is a very strong enhancer. As a result, it can act as a sink for many transcription factors. As a result, the nearby genes often are deficient of transcription factors and their transcription goes down. So simply because of integrating neocassette, you might get phenotype that has nothing to do with the gene you are actually trying to knock out or knock in and the completely artifactual result. To prevent that, nowadays what most of us would prefer, where the neo cassette after selection is done, it is fluxed out. So imagine that the embryonic stem cell, you have selected it for G418 resistance, and then Satyam, So therefore, what is done that after the cell line is selected, then you simply treat it with Cree enzyme, like you can misexpress the Cree enzyme from a plasmid or something like that, and the neo cassette will be fluxed out. Now, the green part is right after your 5' homology arm, where you might have the promoter enhancer, and if the green is itself Cree, then you have a Cree knock-in. Okay, so that's how it is done. The LOXP one, I will come up for it. I will explain it later with the proper construct.
So we are basically using Kree-lock system itself to generate the Kree mouse.
And this is a new variation of the old thing. In the older times, like till say year 2000 or 2003, 4, this LOXP thing was not there. People started to realize the artifactual nature of knockout only around that time. Then they integrated this LOXP. Without the LOXP, also Cree can be knocked in. Okay. But yes, right now, actually, there is another thing. Uh, there is another such recombination system called FLP FRT, where FRT is the site-specific, like the sequence, which is recognized by this enzyme called lipase. So often, Cree knocking would be combined with FRT, FLP, like FRT sites on the other either side of the neo, and that's how it will be taken out. Do you understand?
Yes, sir.
It is exactly like Creloxp, just the enzyme and the flanking sequences are different. It is FLP, FRT. The one thing that I did not talk about like that is, I somehow thought I talked about it and I said questions, and therefore I gave, but some of you have already done your own research and figured it out, but I did not talk about it. So let me see if I can quickly show it to you. Just give me a minute, huh?
Sir,
Uh-huh.
I have a question. I have a question regarding one of the questions that you had shown in the question.
Uh-huh.
We talk about knocking out the gene in the adult pancreas, but not in the
Yeah, so that's what I
pancreas
am
during
showing
the
you.
embryo pancreas.
That's what I am showing you now. That part
Okay, sir.
I did not teach, but some of your classmates were like
Okay.
one person read about this system. So of course, deserves appreciation, and others came up with RNAi. That is also good. But those who failed to see that straight Creloxp will not do the job, I could not give them full credit. But this is what it is.
So
So,
okay.
yeah,
Okay, so I wanted to know if we have to use this Cree ERT system
Yeah,
Cree fused with an estrogen
yeah.
receptor.
So
Okay.
that's what I'm showing.
So that
Yeah. So you know about this cell type specific enhancer followed by Cree, right? Now this will dictate that the Cree is expressed in your cell type of choice. Like it can be cartilage, it can be bone, it can be muscle, whatever. But if you knock out a gene very early on, then the target tissue type might have such deleterious effect that the tissue will not develop properly, which will prevent you from asking the questions regarding adult, like is this gene still important for adult life or not. So for that, what is done is that this a fusion protein is made with the Cree with a mutated version of estrogen receptor. Okay. Now, why mutated version and why estrogen receptor? Estrogen receptor normally, estrogen receptor is a nuclear hormone receptor. It normally stays in the cytosol. Only when it binds to its ligand, that is estrogen, then it moves into the nucleus. So this property is exploited because Kree-Loxp mediated recombination has to happen in the nucleus only, right? Because the chromosome is in the nucleus. So, but the other problem is that normal female mice, they will have estrogen during their estrous cycle, and therefore, even if you don't want to, it can go to the nucleus at any point of time and knockout can happen. So it is a mutated version of estrogen which does not bind to its natural ligand estrogen, rather binds to an artificial ligand, which is a very popular cancer drug called tamoxifen. Now, any point of time in the life cycle of the mouse, you decide that now I want to knock it out, or now I want to do lineage tracing. So you will inject tamoxifen in the intraperitoneal cavity. Intraperitoneal cavity is a big space in the mouse main body. So you inject the underbelly and it will, and you go beyond the abdominal muscle, and then it's a huge cavity, you just inject it in there. The advantage is that it is very quickly taken up by the bloodstream and then it goes to all over the body, including the cell where your Cree is expressed. So there now, because of ERT2, the Cree enzyme will move to the nucleus and recombination will happen. Okay. So that is how we do this knockout, like a stage specific knockout. Okay. Any question?
Sir, can you compare this to RNAi and how which one is better in which case?
See, RNAi, if you want to do it in mouse, you have to do it in one of the two ways. One is that somehow you have to ensure that the RNAi is expressed only when the mouse is in adult stage. Okay. So therefore, you have to make a construct that will be able to make RNAi. Typically, that is done something like this. Sense strand, irrelevant sequence, antisense strand, and then the double stranded of that. And then you have some kind of a construct. Then the RNAi, artificial RNAi will be produced inside the cells and it will do its job in knocking down. But you have to ensure that it is expressed only in the adult life. So again, you will have to take advantage of something like this. injecting RNAi inside of the cells and each and every cell in a tissue type is very, very difficult. Okay, so therefore RNAi is not a very good measure. Like I have given credit only for the thought process, but in reality, it cannot be utilized.
Okay.
Like where you make RNAi constructs in vitro, you know, like this or just this part, and then in cell culture you can actually deliver it. Okay, using lipophilic transfection reagents. But in vivo animals, it is very difficult.
Sir, is it possible that in vivo we can infect the animal with lentivirus containing that shRNA? So
Yeah,
that
it is possible, but lentivirus also does not replicate inside the mouse. Okay,
okay, sir.
so whichever cells will get it, only there the knockdown will happen.
Okay, sir. It will not. They will not get in the particular place. They will get anywhere. Lentivirus.
No, you can, like depending on the tissue, you can ensure that it is injected only in a particular tissue. But the problem is that, say, a tissue has 1,000 cells and your lentivirus infected 100 of them, will you see a phenotype or not is a tricky question.
Okay, sir.
Sir,
Yeah.
what if we introduce this construct into the chromosomal DNA, like something like a Cree knock-in, and then we have like a cell-specific promoter for this construct so that it can be expressed only in adult cells? Can that be done?
Yeah, there are very, very few genes known, if at all, which is expressed only in adult. There are many cell type specific genes, but adult specific enhancer, very few that I know of. Like I know only one in cartilage. That's it.
Okay.
So there the problem comes.
Okay.
And this has been so far a challenge even with the CRISPR-Cas system. CRISPR-Cas system, uh, to program it for conditional change in genomic structure has so far remained a challenge. Whether it is spatially conditional or temporally conditional or both, that's where the challenge is.
Okay.
बट अब सच सच बोलो कि आप में से कोई भी ट्रांसक्रिप्शन वाला स्लाइड्स देखे भी हो कि नहीं? Transcription and transcriptional regulation is the only subject that I actually know. तो वो सीखना है तो पढ़ो.
Sir, I have seen lecture 16. फिर मैंने देखा कि अच्छा lecture 15 भी है.
हाँ.
So lecture 15 is remaining, but lecture 16 I have completed.
वो lecture 16 लेके problem हो गया था. Like I recorded these lectures 2 days back. What happened is that even now the phone calls I am receiving, there is someone in my household whose daughter met with a deadly accident, and the young girl, 26-year-old, she is in hospital and on ventilator. And when I was recording the lecture, suddenly my wife came and informed that no hospital is taking her in if I can help. So that conversation actually got recorded in the lecture. So I had to edit it because these lectures are publicly distributed. So that's why it got delayed in uploading it. And right now I am getting calls because the girl has no hope. So the family will have to decide when to withdraw the ventilation support, and which is painful for the family as well as for us. And because these are very poor people, they do not, they are not educated. So our country's system takes full advantage of that. So I am trying to provide them with some assistance. Pardon? Okay, go ahead. हेलो, और किसी को कुछ पूछना है?
Sir, I had a question from last day's question paper itself. Should I ask?
हाँ.
Okay. So, sir, my first question was about how would you determine the cell type where in a part of mouse eye where ZNMD2 is expressed during all stage of embryonic development and in adults? So, sir, should I apply the process which you taught now? I am not clear about how to figure it, like this question.
फिर से बोलो. I was a little inattentive. फिर से बोलो.
Sure, sir. How would you determine the cell type part of mouse eye where ZNMD2 is expressed during all stage of embryonic development and in adults?
So if you have to look at expression in an animal, what is the method?
Sir, I was thinking of fate mapping.
क्यों? RNA institute hybridization ने क्या गुना किया?
Oh, okay, sir. Yes. Now I understood.
But for I think the eye you have to do a section like section mount, and for the adult you have to do a whole mount.
Whole mount.
Adult तो पक्का section mount करना पड़ेगा because adult में penetration है ही नहीं. For embryo, you see, like Hoimi gave the best answer yesterday, that for any gene, if you want to know where it is expressed, particularly when I clarified in which tissue or cell type, the answer should be that because you do not know where at all, so in the embryo, I'm talking about only in the embryo. In the embryo, the penetration is much better. So you first do a whole mount and get the gross idea of where it is expressed. Then you make sections of those relevant tissues and then do a section in situ hybridization to pinpoint the tissue or cell type.
Perfect. Yeah.
I will show you something that you might find useful.
Okay, sir.
ये आवाज मेरा दिमाग खराब कर दे. Like how bad the noise is for you guys?
Sir, it is like a continuous humming and sound.
It is giving me a headache. And you know, like this is a 1.5 lakh rupee laptop with 3 years warranty, and look the service I am getting.
Sir, is it due to your driver? I think the driver is weak.
I think it is the fan, like either the fan or the hard drive.
Yes.
Can you see it?
Yes, sir.
So in this paper, we conducted RNA in situ hybridization for 1600 genes. It was done at IIT Kanpur only. You see, like these are some of the genes that are extremely specific. You see, this gene is expressed only in the liver, and it does not matter how old the animal grew, it continued to be expressed only in the liver. This one only in the heart. Okay, this one only in the pancreas. But such genes are really rare. Okay, but we found a few. but what I wanted to show you is something different. Yeah. You see, this is a gene that is expressed in the kidney. Okay. From here you can see it is starting to get expressed. Here you see, here you see, here you see it is there. But we got multiple genes with kidney expression. We wanted to know whether all these genes are expressed in the same cell or not. So what we did, we took two different genes. One we labeled fluorescently with red and the other with green. Like RNA in situ hybridization also you can do with fluorescence labeling. And you can see that these two genes are not really expressed in the same cell. Like either there is red or there is green. There is some yellow, but that is most likely artifact. Okay, but while this one, these two genes are absolutely in the same cell. Do you see that?
Yes, sir.
You can only see yellow. So when you want to, like, whole mount hybridization, RNA in situ hybridization can only give you gross reference, okay, in which tissue it is expressed. If you want cellular level resolution, you need to go for section in situ hybridization.
Okay, sir.
Okay.
Yes. Thank you, sir.
Sure. Next. अरे यार. If there is no other question, then we will break off. Is that okay? We will meet again on Wednesday. Is that fine?
Yes,
Sir,
sir.
Yes,
the
sir.
last two lectures you have uploaded are on your second channel. Actually, I just noticed.
Second
Yes,
channel,
sir.
matlab?
It
On
was
the
on the
channel.
channel which you said it was for your personal use.
Yes, that's why, because we could not get the
Oh, so
notification.
can
Yes, sir.
someone teach me how to bring it to the first channel, other channel?
So you have to delete it from that and then
I
upload
think
it.
you'll have to upload it again from
Yeah.
your the channel.
Okay, but okay, so now let me learn something from you guys. This is good. Let me share the screen. Can you see my like internet thing?
Yes, sir. Yes, sir.
Yes, sir.
So okay, see, so this is what I do. You see, I am fairly illiterate about these things. So I go here and then I do this create channel and upload.
Sir, I think like you have logged in from your other account, like the circle, like your profile picture belongs to the other account. I think you'll have to change that and then upload here.
Upload it.
Yes, sir.
It is
Oh.
because you have not changed this
Because
that
when
is
you are
getting
uploading,
uploaded.
sir, it is going to the other personal account.
Yeah.
The
Like you can switch
sign up.
account instead because I
Yes.
think you have the other account logged in.
Yeah.
Yeah.
Oh. Okay.
And now it's in the other account.
So
If you
now if you upload, it will go to this channel.
Oh, thank you so much, guys. This is very, very useful. Now, I do not really remember the user ID password of this other account. Okay, fine. I will do it. Huh? I will do all the three today. Huh? Not to worry.
15, 16, 17.
Huh, huh.
Okay, sir.
17 तक हो गया है.
Okay, okay.
Okay, so I, like without, really sounding pompous, Like, very few subjects I actually know, and of which transcription is the one subject that I actually know. So if you want to learn in depth about transcription, transcriptional regulation, this is your opportunity. And people who will be working in Jayendran's lab, Bushra's lab, any of the genetics, molecular biology type lab, you know not necessarily like Santosh's lab or Divyendu's lab type, but any lab, there your thorough understanding of transcription, transcriptional regulation will be enormously beneficial for the lab. So therefore, take full advantage of it and study this part really well. Okay.
Okay, sir.
Yes,
Okay.
sir.
So, Fid, we are closing it today. Is that fine?
Yes, sir.
Yes, sir.
Okay.
Okay, sir.
Bye. Bye. Bye.
Bye.
Bye.